A genetic disease functions like a factory producing thousands of broken, destructive machines. Traditional pharmacology attempts to solve this on the factory floor, using drugs to wrestle with individual malfunctioning proteins one by one. This approach requires finding a specific physical pocket on a fully folded protein to block its action, a task that is often mathematically impossible for complex structures. This diagram illustrates the therapeutic intervention points within the central dogma, the process where DNA is transcribed into messenger RNA, or mRNA, to build proteins. mRNA is a transient genetic intermediate that carries instructions from the nucleus to the protein-building machinery. RNA interference, or RNAi, intercepts these instructions upstream. It functions as a set of molecular scissors, selectively slicing the blue mRNA strand before the disease-causing protein can ever be manufactured. Researchers utilize a synthetic small interfering RNA, or siRNA, to trigger this process. siRNA is a synthetic double-stranded molecule designed to locate and destroy specific target transcripts. It operates as a programmable search engine that scans the cytoplasm until it registers a perfect match, allowing the cell's own machinery to shred the specific disease-causing mRNA instructions. Biologists Andrew Fire and Craig Mello first documented this mechanism in roundworms in 1998, identifying it as an ancient biological defense system. Their discovery of how double-stranded RNA triggers gene silencing resulted in a Nobel Prize in 2006, one of the fastest validations in the award's history. Because the system relies on the simple logic of matching genetic sequences, it effectively democratizes the human genome. It provides a way to target previously intractable diseases simply by synthesizing an RNA strand that matches the code of the malfunctioning gene. Translating this laboratory breakthrough into a human patient revealed a massive biophysical blockade. Injecting naked synthetic siRNA directly into the human bloodstream results in immediate, total failure. The circulatory system is an aggressively hostile environment. The moment the fragile RNA enters the blood, it is shredded by ubiquitous nuclease enzymes and rapidly filtered out of the body by the kidneys. Even if the molecule survives the blood, its physical structure triggers a lethal alarm. Mammalian cells have evolved to recognize double-stranded RNA as a signature of viral infection. The body's innate immune sensors detect the drug and launch a systemic inflammatory response that can be fatal to the patient. The Nobel-winning discovery is functionally useless in humans without a sophisticated transport vehicle. Success required an engineered solution capable of bypassing 1 billion years of evolutionary immune defenses. By 2010, the pharmaceutical industry reached a breaking point. The delivery problem appeared unsolvable, leading major companies like Roche and Merck to abandon the field entirely in a period known as the RNAi nuclear winter. Alnylam Pharmaceuticals faced an existential financial crisis during this era. They were burning through capital while pioneering the delivery vehicle, eventually logging a cumulative net loss of $2.84 billion before their first drug reached the market. To avoid bankruptcy, the company was forced to execute a massive corporate restructuring. In early 2012, they eliminated 33% of their entire workforce, a move intended to stretch their remaining cash reserves just long enough to reach clinical proof of concept. This corporate desperation created an intense pressure cooker environment. The remaining scientists were left with a singular mandate: engineer a microscopic vehicle that can defeat human biology, or the company dies. The clinical-grade lipid nanoparticle, or LNP, functions as a dense, solid-core nanomachine. It is built from a precise molar ratio of 4 distinct lipid components, each designed to overcome a specific biological hurdle. The first component is the PEG lipid shield. This outer coating creates a steric hydration barrier that prevents the nanoparticles from clumping together during manufacturing and hides them from the body's immune surveillance cells. However, a permanent shield also prevents the drug from entering target cells. Engineers designed the PEG lipid to act as a molecular timer. Its weak chemical anchor allows the shield to naturally shed away in the bloodstream within 2 hours, unmasking the active particle only when it reaches the liver. Beneath the shield, cholesterol and helper lipids stabilize the core. Once the PEG layer is gone, this exposed surface recruits a specific blood protein called ApoE, which normally transports fats through the body. The ApoE protein acts as a Trojan horse disguise, It tricks the liver into thinking the nanoparticle is a natural fat molecule, allowing it to slip through the microscopic fenestrated pores, or sieve plates, that line the hepatic blood vessels. A liver cell programmed to absorb fats swallows the disguised nanoparticle via endocytosis. It traps the LNP inside an internal cellular vesicle called an endosome, which the cell begins to acidify to digest the contents. The final component is the ionizable cationic lipid. In the acidic endosome, this lipid acquires a positive charge and shifts into a cone-like structure. This shape change violently disrupts the endosomal membrane, tearing it open to release the RNA payload into the cytoplasm. By precisely exploiting the body's own transport proteins and its cellular acidification process, the engineers successfully smuggled the genetic kill switch into the heart of the cell. This 16-year engineering gamble culminated in the 2018 FDA approval of patisiran. It was the first RNA interference therapeutic ever cleared for clinical use. The targeted LNP delivery system successfully shut down the production of mutant proteins in the liver, halting the progression of a previously undruggable and fatal genetic disease. Commercializing this technology required scaling the complex fluid dynamics of microfluidic mixing. It meant moving from tiny laboratory batches to commercial multi-kilogram production while ensuring every nanoparticle was uniform and stable. The legacy of this delivery struggle extends far beyond liver disease. The same lipid nanoparticle architecture perfected during the development of RNAi became the indispensable delivery foundation for the mRNA vaccines used globally to fight the COVID-19 pandemic. Breakthroughs like RNAi and mRNA are rarely just discovered in a laboratory. They must be painstakingly engineered to overcome the harsh realities of human biology.